talks today. Andy Smith, who is was a PhD student here at MIT and a postdoc in Zurich for a while, and now he's in Leipzig, and he's going to be telling us about molecular dynamics calculations and integrating those with NMR. And the second speaker is Professor Lynn Mueller from UC Riverside, and he's going to be dealing with non-uniform sampling experiments in the second presentation. So Andy, the virtual floor is yours. All right.
Okay, well, thanks, Bob, for the kind introduction. And let me see if I can get my screen up. Okay. Okay, do we look okay?
I don't know if you could hear me. I wanted to apologize for coming in late. I had just a avatar of myself, but I had something important to do before I could join you guys. So this is Alex Pine saying, hi, welcome, happy to be here. I'm reminded of Groucho Marx's comment, which was, he said, before I give my talk, I'd like to say something important. So I have something important to do before the talk, guys.
All right, okay, Andy.
Good to see you.
Yeah.
Okay. Thanks. Okay, and yeah, so also I'll just quickly say thanks to both Bob and Kong for organizing this. It's nice to be able to share research and see some of the research while we can't go to normal conferences. Anyway, today I'm going to talk about what we're calling the dynamic landscape and how we can characterize it using NMR simulation, NMR experiments and MD simulations. And so first of all,
you share your screen, Andy.
Oh, I'm sorry. I started to do that and then I stopped it.
Just
okay,
remember,
let me.
Yeah, click. Remember to tick the box like with the video.
Yeah. Yeah. Okay. Okay. It's there now, right?
Yeah. There you go.
Sorry, the Zoom window goes away when I go into presentation mode, which is a little weird. Okay. Anyway, here we are. Yeah. So, well, I guess the question is, what is the dynamic landscape? That's our starting point. With NMR, we're sensitive, of course, to, among other things, reorientational dynamics. And this is via modulation of any anisotropic tensor. Today, we'll focus mostly on dipolar couplings. We'll look at motion of HC bonds, and so we'll effectively be looking at the motion of the proton-carbon dipole coupling. But this kind of approach also works with quadruple couplings, also works with chemical shift anisotropy. So what is the dynamic landscape? Well, it's all the factors coming in to affect that modulation of the couplings and effectively describe the motion of a proton-carbon bond. And so the first thing that comes in is just the orientations we sample. And so this is just a cartoon, but you might sample a few different orientations, and all those orientations sampled are going to lead to an order parameter, or here we'll use the generalized order parameter S squared. And so as the motion gets larger, S squared gets smaller. Of course, I'm going to plot 1 minus S squared. I like this parameter better because when motion gets bigger, the parameter gets bigger. But that's just a small part of the story. The motion also has a speed. It goes at a certain rate. We'll describe that with the correlation time. And here we say, well, okay, so let's look at one bond and one motion. And so we say, well, we've got an amplitude, we've got a correlation time, and potentially we've got a distribution of correlation times. And so today we're going to characterize that distribution also with a width. But again, it gets more complicated because there's no reason that you're looking at just one motion. Anything that rotates your HC bond is going to influence your measurable parameters, so your relaxation experiments. And so you might have motion 1 with amplitude and a width and a correlation time. And then you can start adding these together. You get motion 2, and then you might have a third motion, and eventually you get this complex distribution. In my scenario here, I've got 4 motions with 3 parameters each, and so 12 parameters. And you start to imagine why I'm talking about doing this with simulation and experiment, because that gets to be a lot of parameters to fit just based on experimental data. And then finally, the whole landscape is this idea, well, that those distributions against correlation time are going to change as a function of position in your system. And so the question today is, well, how much of this, what we call the dynamic landscape, can we characterize based on the combination of molecular dynamics, simulation, and NMR experiments? And so to demonstrate the approach that we're proposing, I'm going to use POPC bilayers. And so why we chose these? Well, first of all, they're highly dynamic. All positions on the molecule have a very high amplitude of motion. Those motions are distributed over multiple orders of magnitude, so multiple timescales. And they're also functionally important. I mean, they're a critical barrier in cells. Obviously, you're going to need order, or else they're not going to act as a barrier, but you also need disorder. You have lots of proteins embedded in these bilayers, and if the bilayer is too rigid, the protein's not going to have the flexibility it needs to sample different conformations that allow it to function. Now, hopefully you're all seeing a video on the right, and it's just a POPC molecule extracted out of an MD simulation. And what I'm doing with it is I'm just simply log spacing the correlation time. And so like right now, over the course of 1 second, you'll see about 50 picoseconds of the MD simulation. And by accelerating that, you just can see how the motion changes as a function of time scale. And so we're going to use this type of video to view a few different things out of the MD simulations. If we're having any trouble, you'll notice I typed into the chat window a link to YouTube, and all the videos that are in the talk are also in this YouTube playlist. So you could also check it there. And you can also go back to it later if you want to see the videos again. Okay, so here's our strategy for characterizing the dynamic landscape. Now, the first thing we're going to do is we're going to take a series of NMR experiments, and we're going to take an MD simulation, and we're going to cross-validate these things. So we're going to come up with a parameter. This is going to be a detector analysis, which we've come up with the last couple of years. And I'll describe that briefly. But we're going to basically show you that the motion in the NMR is pretty similar to the motion in the MD simulation, at least enough so that we can proceed to use the two methods together. Now, the next thing we're going to do is, well, we said that there's a very large number of parameters to describe the dynamics even of just a single bond. So what we're going to do is take the MD and we're going to use it to separate the motions into components. And the argument is that while the whole motion might be described with up to on the order of 10 parameters, the individual components we hope we can reasonably characterize with just a few parameters. And so here will turn out to be about 3 parameters. From that, we can build up a full landscape from the simulation. And then finally, we're going to go back to our experiment and we're going to refine the landscape based on our experimental data. And so we start with the experimental data. We focus on 2 types of experiments. First of all, you can measure residual tensors with NMR. I mean, everybody on the screen knows this already. But anyway, you measure the rigid coupling. So if the molecule's not moving, you know how big the dipole coupling should be between a 1-bond proton-carbon bond. And you can measure the residual coupling and you get an order parameter. Now there's a couple points. First of all, the order parameter is the result of all the motions rotating the given bond. And it's all motion basically within the NMR timescale. You could sort of define a window. You could say the order parameter is everything in this range, and then at some point this gets cut off. And so here I'm doing 1 minus s squared because this is more related to the relaxation, but ultimately, this is the familiar order parameter. So that's one experiment we can do, but the other one we can do is relaxation measurements. In this project, we'll look at T1 relaxation of the carbons. We'll look at proton-carbon heteronuclear NOE, and we'll also look at T1 rho. And so if the order parameter kind of captures all the motion within the NMR timescale, I'd like you to think of the relaxation as kind of a window into the total motion. And so this would be the window from T1 at 600 megahertz, carbon 13 T1. And so you see the idea here is this, it just captures everything kind of around a nanosecond with a width over a couple orders of magnitude. So if a relaxation experiment is just a window into the total motion, kind of a way of filtering with time scale, then what a detector is it's just a better window. So in this case, we take 7 relaxation experiments. We also measure the dipolar couplings to get the order parameter. So we have a total of 8 experiments. And we recombine these experiments to make better windows. So if you look, if I say which one of these things is a window, well, they're kind of broad and they're pretty badly overlapping. The new detectors are now much more separated and also significantly narrower. So you get that window. It's well defined. And I mean, I give you the functional form down here, but I don't want to spend too much time exactly on the functional form. But it's just capturing all the dynamics in this range. Now, the reason this is powerful if you want to work with multiple methods, and particularly with molecular dynamics simulation, is, well, from MD, you can calculate the correlation function itself. And it turns out that if I take a time point in the correlation function from the MD, that's basically just another window. It's a different shaped window, but we can optimize them to match the detector windows we use for our experiments. And so we get a parameter that's very comparable from NMR to MD. And so we'll use that to validate the MD simulations against the experiment. So this is just a summary slide of our experiments. We do proton carbon NOE, we do carbon T1 at 400, 600, and 700 megahertz. And we do R1 row at 3 different spin lock strengths. We don't end up using these R1 row experiments that much in this talk, but nonetheless, we worked with those experiments and we do a dip shift measurement to get the order parameter. This is our detector analysis. It'll become a little bit clearer what this means in the next slide. But you see, we're not fully resolved on the POPC. We did this experiments in 1D, but we can make that up with the MD simulation. And so that's what happens here. We do this detector analysis, and we have these windows from the experiment. Those are the colored windows over here. These 2 detectors are sensitive to like multiple microseconds of motion, and so we're not going to try to reproduce those with the MD because the simulation is not long enough. We have about 8 microseconds of simulation. But the other ones we try to reproduce with the MD. The black lines are the MD-derived detector windows, and you see they match the experimental windows. And these are the responses. And so you can compare the black lines to the colored lines, the black being the MD, the colored being the experiment. And you see that we're overall in pretty good agreement. We're not in perfect agreement. We have a particular disagreement here in the head group. So you can map this down to the molecule here. There's some disagreement in the head group. This kind of implies that the head group motion in the MD is too slow. But the other thing I can do is, well, okay, so I have good agreement in the chains. I don't have experimental resolution. So what I do is I say, well, if the MD agrees with the experiment, I'll just take the MD results and I'm going to plot them on the POPC molecule. Where I don't have as good agreement, I'm going to take the experimental results and I plot those on the molecule too, and I get an overall picture. And so you see what's happening. I mean, you see the overall motion. You see that motion at the ends of the chains in POPC is very fast and very large amplitude, also in the head group. You see a sharp contrast to that. So a lot of this motion is basically less than 100 picoseconds. I guess I should take a step back, by the way. You get motion at 100 picoseconds, 800 picoseconds, 4 nanoseconds, and then also here in the microsecond range. You've got this sort of weird detector row 0, which catches everything. Oops. I can't get out now. Okay, there we go. Catches everything else. Now I can check in the MD and I can tell you that almost all of this motion in row 0 is less than 100 picoseconds. That's why I say that's less than 100 picoseconds. In reality, a little bit of the glycerol backbone here is coming from this part of the window here. But But for the most part, we say this is fast motion. Okay, so anyway, fast motion, less than 100 picoseconds in the head group, and in the ends of the chains. In the backbone is much, much slower now, happening around 4 nanoseconds. So you see a really big contrast. And if you want to see, if you really want to get a grip on this motion, well, what I've done is I've plotted those detector responses onto an extract from the MD simulation. And so at the very short correlation times, so when we're taking very short steps, well, you see big steps in the ends of the chains and in the head group. Now, as we start to increase our time scale to longer correlation times, now you start to really see the detector responses are coming up here in the backbone, and that's starting to happen when really the whole molecule starts to move around. And just to give you a second chance to catch all that. See, here you have large steps almost immediately over the course of just a few picoseconds. Whereas if you were to look in the backbone, you see that the reorientation is very small comparatively. Whereas once we get to the nanosecond range, now this reorientation in the backbone gets quite large. So far we've done fairly well. We've combined our experiment simulation to get full resolution. We get a video that gives us a hint what our experimental results mean, but we'd like to go another step. And what we try to do here is we'd like to separate our motion into components. So if you consider a bond vector, I mean, a bond vector time tau, to achieve its orientation at some later time, t plus tau, well, we just apply a rotation to it. And that's just a rotation matrix. Well, arguably, we can say that that rotation matrix is the product of several individual rotations. And so that's what we do. We say, well, we've got a product of individual rotations that leads to the total rotation of the bond vector. And in special cases, if we pick these rotations well, each one of those rotations can be associated with a correlation function, so that the total correlation function is the product of the individual correlation functions. And this is going to work well if the two motions, if the motions are statistically independent from each other. And in special cases, we require that they're time scale separated. And you'll have to probably ask me when that is if we want to get back to that. Okay, and so the way we've implemented this is something called the frame analysis. And so to separate into multiple motions, you're going to need multiple frames, but I'm just going to talk how this works for a single frame. And so what you do is you define a frame, and that frame allows you to separate motion in the frame from motion of the frame. Just a simple example, let's say I've got a methyl group rotating, and so the total motion of an HC bond in the methyl group is the result of, well, the methyl rotation itself, but also the result of reorientation of the carbon-carbon bond to the methyl group. And so to define a frame, I just take a frame where the carbon-carbon bond stays fixed along Z. And if I do this, I get just the methyl rotation, and obviously I've removed the carbon-carbon motion. So that's motion in the frame. And so I figure out, well, what are the rotation matrices for that? And from that, I can extract a correlation function. Now, the hard step in this is that additionally, we calculate, well, the correlation function for motion in the frame, we calculate the residual dipole tensor for motion in the frame. And then we apply the motion of the carbon-carbon bond to that residual tensor. And by doing that, we are also able to get a second correlation function, motion of the frame. And we can do this iteratively to separate out multiple motions. And so that's what we've done here. We've taken the POPC dynamics from the MD, and we first separated out very local motions. We looked at one bond librations. And the idea here is we just take an HC bond, and we also take all the other atoms bound to the carbon, and we align these 5 atoms to a reference structure. And then we look at the motion within that structure, and that gives us basically just a local distortion, basically the HC libration. It's a little easier to understand this if I jump the whole way to the other side. If we look at the overall motion, so I separate this, the molecule into 3 parts. I take the glycerol backbone and the oxygens of the glycerol, and I again do a RMS alignment. and I apply that frame to the whole head group and backbone region. And so looking at that, I can get overall motion of the head group and the backbone. And then on the other side, I can get motion within the head group and backbone. So I get internal motion. And I do the same thing with the chains, except here I take the moment of inertia of each chain. I take the longest component, which basically is a vector that's going to point in the direction of the chain. And that's going to define my overall chain motion. And once again, I can have internal motion. So here you get the overall motion. And then here in these two plots, you have the internal motion minus the librations. I've done one more separation. So in the head group and on the carbonyls and at the double bonded position, there's just one internal motion, but everywhere else in the chains, I separate this into a perpendicular component that rotates around the moment of inertia and a parallel component that kind of moves up and down on it. Okay, and so you can see that once in action. I mean, you'll see that the librations don't do hardly anything. And by the way, these are orders of magnitude smaller than all the rest of the motions. You can see the internal motions. These are both relatively fast, occurring either under 100 picoseconds when it's blue, or around 100 picoseconds when it's orange. It's the overall motion that's much slower, and also, and we'll have to wait till the video repeats, the overall motion is spread pretty broadly. So you see a big detector response both at short correlation times, but it stays at long correlation times. And here the internal motion, so this is perpendicular and head group motion. So the internal motion of the head group, you see actually that the glycerols in the head group are some of the slowest moving things here as far as internal motion goes. And then you can see that's also reconstructed over here as the overall motion. So that's one of the things we can do. The other thing that I think is worth taking a quick look at is the residual tensors from each motion. These are, of course, important to the analysis because when I calculate motion of a given frame, I need to apply that motion to the residual tensor of the inner frames. So all the motion within that frame. And so you can see what the different things do here. Libration, I mean, we already established on the previous slides, this is very small, and so it hardly averages the tensor at all. This is the time trajectory. Parallel motion has some small averaging effect and creates a small anisotropy. Other internal motion can have, in the head group in particular, basically just makes the tensor go to zero entirely. What I think is interesting is you have this rotation around the moment of inertia. And well, it's interesting because it's exactly what you expect, is that all the tensors realign themselves so they go along the moment of inertia. And so that's what you see happening here. And then the overall motion, so the overall head group motion and chain motions just lead to some tensor averaging. And of course, you get to see a video of this in action too. And so you can see like these are quickly reorienting along the chain moment of inertia. Whereas if you look down here, for example, the overall motion is taking much longer for these tensors to disappear. Like there goes the overall motion of the head group and the backbone, and these also start to get averaged out. Okay, so we've got now our motion separated into frames. And the idea of doing that, one of the big ideas of doing that is that now the motion should be simpler. We said the total motion has on the order of 10 parameters in it. But the motion of each frame, we say potentially we can parameterize with a few parameters. And so that's what we do here. Each motion we characterize with an amplitude, 1 minus s squared, a correlation time, and a width. By the way, the librations, we don't really have, they're just really fast in the MD, so we don't really get the width out. I just fix the width. But the rest of these, all three parameters float. And so these are the parameters you get. These are the calculation of the detector windows. By the way, to do this step, I use a higher resolution detector window for the MD. It's not directly comparable to the experiments, but it's perfectly good for analyzing the MD and gives you kind of a more certain result. And you get a landscape. So for each one of these motions, you get a 3D plot that looks like this. And so you really start to see like the details of all these motions, of the individual motions. And finally, you sort of take, well, you need to take the product of the correlation functions themselves. This is not that difficult of an operation, but it's ugly on a slide. Anyway, we take the product of all these motions and we get the total landscape, which is already pretty good. But keep in mind that this is mostly MD derived. Now, we did validate it in the first step. We said the MD is doing pretty well. But I can do a couple things. First of all, I can calculate, and this is one of the great things about this detector analysis is you can always compare one thing to another. I can calculate detector responses from my landscape. And so that's what I'm doing. The blue line here is, again, the experiment. The gray dashed line is the landscape simulation. And so I can check how well am I doing here. I'm doing fairly well, but there's definitely some disagreements. And so in the last step, I can refine my landscape, my parameters for my landscape based on the experiment. And so that's the new black dotted line here, simulation plus experiment. You see that my agreement improves. I've improved my agreement by adjusting one parameter per experimental resonance. And so that parameter is basically a scaling factor in the correlation times. I move, for the most part, I increase the internal correlation times. Now you may be saying, well, wait a minute, you've changed something in 2 plots here, isn't that 2 parameters? But they're scaled equally. So they're scaled by a multiplication factor. And so the result of that is actually just a single parameter per experimental resonance. And it's scaled also if they're overlapping resonances, then I scale all corresponding resonances equally. And so finally, we get our landscape that's based on a parameterization from MD, but it's actually finally refined using our experimental detector responses. And so that wraps up what we're doing here. We get a comprehensive characterization of a dynamic system. We first used our detectors for comparison of the experiment and simulation. So we checked that the simulation is high quality. We simplify our problem by separating the motion out in the MD simulation. We parameterize it, we combine it to get a new landscape, and finally we do a refinement step, going back to the experiment. And I should mention that, the PLPC has been pretty well studied, although we do, I would say, separate out some motions overlapping in time scale and really get the whole picture at once. But this is also a totally general approach. I mean, the things you need are good experimental and simulation data, and you need somehow separable motion. So you need to be able to define frames that actually separate out your motion. And so just to thank a couple people, this project's been mostly done at the University of Leipzig with Daniel Huster, and Peter Hildebrand's been supplying our MD simulations along with Alexander Vogel. Oscar Engberg did the sample prep. I should also mention the detector development all happened at the ETH. Also with Fabian, has done solution state NMR with me, but actually a lot of the mathematics we had to develop to deal with his field cycling experiments get applied to the MD simulations. And finally, this is Kai, he's just started with me, but when you finally get to use the software and it's a user-friendly software, that's what he's working on. Okay, so that concludes my talk. And thanks for your attention. Let's see if I can.
Hey, that's Emily.
Huh?
That's Emily. No, don't worry about that.
Emily?
All right. Yeah. Okay. Whatever. You can keep your screen, or I'm just gonna ask the question. So thank you very much for your talk, and thanks for showing us how you use the detector analysis to disentangle different components. I thought that it was quite cool that you can actually do that. And while actually Alex raised his hand. So Alex, would you like to ask a question or comment?
Digital hand. Nice talk, Andy. Thank you very much. Beautiful. real applications. You made one comment at the end. I just wanted to clarify. I think I understand it. You said field cycling. I can understand how that comes into the context of your considerations. Field cycling as considered through the molecular dynamics. So I understand you're doing field cycling, but it's in the simulation. You haven't done field cycling experiments. We're not basing it on any field cycling experiments out there. Or am I wrong about that?
Oh, no, I should be a little bit careful. What I meant by the comment is simply, this is a separate project with Fabien Farage. We've used the detector analysis on field cycling experiments. And so he's not worked directly on this project, but the mathematics we had to develop to deal with the large data sets for field cycling end up applied directly to the molecular dynamics simulation. But
so field cycling with
maybe like actually like maybe like I will ask my colleague, like, because we have a setup here. Fabian, would you like to comment on Alex's question?
Well, we have worked on protein dynamics with Andy. So everything that you showed here, I was absolutely not involved in, but very happy to discover. But clearly, I mean, there is a lot of field cycling that has been done by Al Redfield and Mary Roberts on lipid, and by looking at Andy's presentation, I was really thinking that that would be a really nice thing to explore.
I agree. I agree that the experimental element enlarges hugely the possibility of correlations, that is looking at correlation times of different fields. You're doing actually correlations of motions induced in the, for example, in the solvent, one picks up motions of parts of the protein and the whole protein. So that dynamic simulation is terrific, but it'd be very nice to look at Fabien's work and others in this field to do experiments and to correlate those experiments with your very nice simulations. So I agree with Fabien, that's worth looking at. Thanks, Andy.
Yeah, thanks.
Okay, I'll read out the first question asked by Anne McDermott. The hierarchy of motion was elegantly presented. I think that you're assuming independence of the various levels. Can you comment about this? I mean, correlation functions, and I think you assume that they are independent and the time scales must be very different.
Yeah, and so it does get, so we assume they're independent. We don't have to assume, let's see if I can explain this. We need definitely statistical independence, and we usually assume that this isn't going to be perfect, but we basically back calculate the correlation function and compare it and see how well we do. And so we did a frame screening step where we tried lots of different frames and saw what gave us the best results. And so the combination of frames I show you here gave you the best results. We require timescale separation if the tensor changes shape or the tensor changes orientation within some frame. So like that's what's happening here in the perpendicular motion, the tensor really changes orientation and shape. If this reorientation were overlapping heavily with the overall motion, we would have a real problem. Because then we don't know, this overall motion has to get applied to the residual tensor of the internal motion. And so if the two things happen on the same time scale, we don't have a residual tensor to apply it to. So that's where the time scale separation really comes in. And it's not perfect. I mean, so I will show you the validation, and I mean, you'll see it's not perfect. So what we do here is we calculate the product of our correlation functions and perform a detector analysis. That's the black lines, and then we do a direct calculation, which is the colored lines. And the form is retained, but we do get a little bit of disagreement. And there's two explanations for that. One is that the overall motion it actually all has to do with the overall motion. First of all, the overall motion shows some really fast components. And that creates first a problem that for those very fast components, we're probably using the wrong tensor shape, right? Because the tensor is reorienting, especially due to that perpendicular motion, that rotational motion. And so we don't quite get the tensor shape right. And then the second question is that fast motion real? When you say that when you say that the frames have to be statistically independent, actually, one of the main reasons they're not statistically independent is you've defined a frame that doesn't follow the real motion. So with my moment of inertia tensor, if the bottom of the chain moves a little bit, well, it moves the moment of inertia tensor, but that doesn't mean that the top of the chain moved with it. And so at the top of the chain, I've got a frame that's moving and it's not really moving the bonds. And so that's also going to create some error. And so we're kind of, so that's kind of the reasons that this is not a perfect agreement. Now, I would say that, yeah, this is doing really well. And also because I've tried a bunch of other frames that I say that we're doing really very well as far as separating the motion. But yeah, so that's kind of where these things come in and where these affect how this kind of analysis works. It also probably can be improved more. I mean, the frame definitions are flexible and we keep trying to update them, but at some point we have to wrap up this stage of the project.
Yeah, I think like what you just say, like it's actually like it partially answers Lauren and just question, like because he asked like, oh, can you do other frames? Can you find the frames automatically for a protein?
Yeah, no, we can't find them automatically, but we sort of implement a library of good choices. So, for a protein, I mean, the peptide plane, librations out of the peptide plane is a really obvious frame, and that you just get from RMS alignment of the plane. If you've got alpha helices or beta sheets, realignment of the whole alpha helix and beta sheet is probably another, and just an RMS alignment of that is probably a really good choice. And we've done a little bit of this on MD simulations, and it seems to work, but we work on keep improving that. And then you get, if you've got a membrane protein, certainly overall motion also makes sense, right? To align the whole protein as a frame, because it might be rotating in the membrane, although it depends how long your MD simulation is if you're going to capture much of that.
Yeah, and also like a couple of people asked, like, can you like elaborate more on like this detector approach? Like, how is it different from the spectral density mapping? And
yeah, okay, and so spectral density mapping is a great comparison. I mean, if you know that, because spectral density mapping is like a linear recombination of your experimental data, and detector analysis is a linear recombination of your experimental data. Spectral density mapping is a general is other way around. Spectral density mapping is a special case of detectors. Of course, it was there long before detectors, so maybe it's not quite fair to call it that, but yeah, so it's just this linear recombination. That's why I sort of show you here is you take each one of your experiments and add it together with different parameters. And so like these 4 experiments add up to this detector. What was the second part of the question? It was compared to spectral density mapping.
oh yeah, it was the same question. It's like he was just asking you to elaborate more on the detector approach that you published on Unveiling, and because like I'm assuming like detector approach is like, I mean, many people are not familiar with it, so like, we're just asking you to elaborate more on it.
Yeah, okay. I mean, the simplest way to understand it really is that each experiment, and from a mathematical side, we say each experiment is a sum of all the contributions from all the motions in the distribution of correlation time. So you take this thing, you say, okay, well, this is my total amplitude, 1 minus s squared. And you say theta here tells me how this thing's distributed as a function of correlation time. I take z to be the log of the correlation time. And it just sums it up where this thing are these windows here. So there's a weighting factor depending on the experiment you choose. And you can just take your experiments, you just add them together with a different weighting. And if you work that out, you just get a new function. And so the trick is to optimize the new function to give me nice windows. And it gets a little bit more involved in that. We do some things with the noise, the standard deviation of the experiments to give a more stable analysis. But that's the heart of the idea, is that we just take our windows from our relaxation experiments and make them better. So we get a higher timescale resolution. And you can do the same thing with MD. These are the MD windows. Each correlation, each delay time in your correlation function gives you a different window, and you can use these and you match them up to the experimental result. And so it makes a really nice parameter for validating an MD simulation.
Yep. Okay. I'll just go to the last question by Walt Masevsky. I also had the same question. Like when you did your MD simulation, was it performed on only one POPC molecule or multiple POPC molecules? I mean, like how would including them, like the molecule interaction affect your analysis?
Oh yeah, no, it's on all the simulations are, the simulations I showed you are on 256 molecules. And so it's a bilayer. So there's 128 on each side. And then there's also 50% water. So half and half on each side. And so, yeah, I mean, no, the MD is not going to work without the whole layer. In this case, we also did some larger simulations. So we did 1,024 POPC molecules and 4,096. And for the time scales you see here, they really don't change much. I can't remember if I put the slide in or not. I think I didn't. But anyway, for these time scales, it really doesn't change much. When it gets out into the hundreds of nanoseconds, they start to disagree. But I'm not even sure if that's not just noise, as opposed to, because you don't have that many events. If you have 5 microseconds of simulation and you look at 100 nanoseconds, you only have 50 events. So you start to have influence of just noise, as opposed to physical differences. We think there should be eventually changes in like due to the long range motions, like collective motions, where you have certain wavelengths available based on how big your simulation is. So yeah, the size of the simulation matters immensely to the interactions, although it doesn't matter that much when you go from something like 256 to 1,024.
All right, so it starts converging at 256 roughly.
We didn't try any smaller than that, so maybe it converges before that. I mean, it's when it gets bigger, you're going to be able to capture more long range motions. I mean, you can't have a motion longer range than the size of your box in the simulation. But when you're looking at the experiment, you're not capturing very well multiple microsecond motions, and so maybe those wavelengths just aren't coming in yet. So. Okay. You need both a really long simulation.
We have our last question from our good friend. If there's repeat Agawa that we know, yeah, how do inherent errors in experimental measurements propagate in your detector approach?
It's basically a linear analysis. So you can use usual propagation of error. So it's just you've got these. You add your experiments together with different weightings, and so you just take the standard deviations you take the variances and add them together with the same weightings. And then if you want the standard deviation at the end, you just take the square root. If you really want to be critical, we do put some bounds on the size of the detectors, which can change the error slightly. And then you have to do something a little bit fancier like Monte Carlo. But in most cases, I get the same answer when I do Monte Carlo and when I just do simple propagation of error. So, okay,
thank you for answering all the questions. Bob, you have anything to say, add on?
Well, I was going to comment that there's a huge data bank on dynamics of lipid molecules from deuterium NMR, and you really haven't. You didn't really seem to address that data. I mean, that collection of data. For example, it would be interesting, I think, to do. For example, you can measure the T1 anisotropy of the deuterium powder patterns, and we did some of that, long time ago now. And as you go down the lipid chain, you see what we call the positive anisotropy. So the parallel edges relax back before the perpendicular, and then it goes to 0, where they relax together. And then you get a reversal where the perpendicular edge relaxes back before the parallel edge as you go down the lipid chain. All right. Now, this was in a glycolipid. It wasn't POPC, so it's a glycolipid. It's a different beast than what you're dealing with, but there's really a lot of other data out there that would be interesting for you. I'll send you some papers, and maybe we could talk about it. All right.
Yeah, perfect. Sounds good.
Yeah,
Thanks,
yeah.
Bob.
Okay, so let's go on to the next talk. Our second speaker is Lynn Mueller from UC Riverside, And Lynn is going to tell us today about non-uniform sampling and how you preserve spectral knowledge in the time and frequency domains. So Lynn.
Okay. Yep.
Okay, go ahead.
Great. Well, thanks, Bob. It's a pleasure to be here at Bob and Kong's Perpetual Online Conference. And so thanks both for putting these together and keeping the community together when we can't be physically together. So today I want to tell you about non-uniform sampling and the preservation of spectral knowledge in the time and frequency domains. I'm going to focus on data acquisition and processing, Monte Carlo simulations, and non-uniform sampling. And I have to admit up front that when I first heard about Monte Carlo simulations, I was a bit of a quiet skeptic. It's not that I didn't believe my friends who kept telling me how great NUS was. It's just that I knew there were some non-obvious aspects of signal to noise and data processing that made me sort of a skeptic about most claims. But I'm not one anymore, and it's Monte Carlo simulations that have made me a true believer. So let's start with just one quick fun example to show you why I was a bit skeptical at first. And so if I start with these two spectra, and I asked the first year graduate students, which one of these is better? They had no doubt, take this one over here on the left. They wouldn't be entirely correct there because it's a trick question, because from the quality of the spectral parameters that I can extract from these, they actually are entirely equivalent. Now, clearly, they don't have the same signal to noise in the Ernst sense of peak height divided by the RMS noise. And so to understand why they're equivalent, I want to introduce this alternate definition of sensitivity as spectral knowledge. And this definition is really more in line with what we really care about when we look at data, which is what's the knowledge or certainty of the extracted spectral parameter that you can get out of it. So first I'm going to introduce the coefficient of variation. This is the error in an extracted parameter sigma divided by its expectation value. And of course, sigma is what we really care about because it tells us if we repeat this experiment, how close would our extracted parameter be to a previous measurement or hopefully to the actual measurement. Then I also want to introduce the knowledge. And of course, knowledge is the opposite of uncertainty. So we'll define it as the mathematical inverse. In straightforward cases, you can determine the standard error by just calculating the covariance matrix. But we have to be really careful in NMR because most of the processing that we do in NMR introduces correlated noise. And that's a real showstopper for simple calculations of the standard error because you can't just look at the spectrum and get all the parameters you need. So rather, we're going to use a much more general method, Monte Carlo simulations. And this method is incredibly robust and it has a really nice intuitive feel and correspondence to our actual experiments. So it's easy to intuit what these parameters actually mean. Okay, in Monte Carlo simulations, we're going to start with a synthetic signal that we know. We're going to add noise to it, chosen from a well-characterized distribution, typically Gaussian white noise. And that's going to give us a model signal. And then we can fit that model signal in the time domain and get out the signal amplitude, the frequency, and the spectral line width. Or we can pre-process this signal, we can Fourier transform it, we can post-process it, and then fit it in the frequency domain to extract out those three spectral parameters as well. And then we're going to repeat this 10,000 times with new draws from the noise distribution, keeping track of the fit parameters that we just histogram up at the end. And from that histogram, we can get out the expectation value, the mean, mu, and the standard error sigma. And again, this really corresponds nicely to what we mean in terms of an error in a measurement, because it tells us if we keep doing this experiment, how close am I going to be to that average or true value? Okay, so let's return to this first example. What I didn't tell you about this is that these spectra come from otherwise identical signals, except for in the second case here, I forgot to turn off the receiver. And I just acquired a FID that is 16 times longer than in the green case. So this is a direct acquisition example where I don't really pay a penalty for longer acquisitions. I just have to pay a probe bill. And the question is, does this really make the knowledge of the spectral parameter worse? So let's see, I can Fourier transform these, I can fit them, and I can repeat this 10,000 times and histogram up the results. And from these, I get the mean and the standard deviations. And I can plot these as a function of the acquisition time, Tmax. And that's what I'm showing here. And what you get is sort of surprising. It's not what your instructors told you would find. What you find is that after you acquire out to about 3T2, the knowledge of the parameter plateaus and it doesn't get any worse. It stays constant, even though the spectra start to look worse. And so the reason this happens is that although the spectrum on the right has 4 times the noise, there's also 16 times the number of points defining the curve. And as all of us know who have whittled away the best years of our lives watching the signal to noise grow in at the spectrometer, knowledge scales with the square root of the number of the measurements. And so these two completely compensate. In fact, the general result is that the knowledge of any of those spectral parameters is simply proportional to the signal to noise ratio times the density of the independent points in the frequency domain. Now we do have to be careful. I need to say this is the fundamental or intrinsic signal to noise ratio, not the apparent signal to noise ratio that you can change by, say, apodization. So we have to be a little bit careful what we mean by signal to noise there. Okay, you can get some additional insight into this correspondence by noting the equivalence of fitting in the time and frequency domains. And here what I'm comparing is the knowledge of the signal area or the amplitude as a function of Tmax when I fit them both in the time domain as well as fitting them in the frequency domain. And of course, it's an important property of the Fourier transform that it preserves information content. So these errors are the same in both dimensions, provided that we actually use the correct formula in the frequency domain for the FT pair of a time truncated decaying exponential. And that equation is shown here. We solved for this in our JPCA article. And for those of you who are interested in this sort of thing, Colin Smith's group has recently worked out an even larger number of interesting line shapes under various apodization functions in his JMR 2020 paper. Okay, so when you look at this in the time domain, it's actually quite obvious why the signal acquisition doesn't matter after a certain cutoff, because the noise points out here are just randomly spread about 0, and they don't influence the overall fit. In other words, not all noise in this free induction decay is equivalent. It doesn't affect the time domain or the frequency domain fit as much as noise in the early parts of the FID do. And that shares some of the ideas of non-uniform sampling that we'll return to in a bit. Okay, so to talk about non-uniform sampling, I need to switch to the indirect dimension. And this has different considerations than the direct dimensions because each point adds linearly to the overall experiment time. So the relevant question to ask is, for a given number of samples, how should they be arranged for optimum parameter knowledge? We can do uniform sampling with a constant time step or dwell delta t out to say 3T2. That would have this sampling density up here. We might decide to truncate acquisition just at 1.5 T2. Or we can use a variable sampling rate with a non-constant dwell that may bias the samples towards the initial part of the FID where the signal is stronger. And for example, we might choose an exponentially biased sampling schedule in which the distribution falls off with a decay constant that matches the signal envelope. And this is called match sampling with a bias of 1. Or we can use a more gentle bias where we fall off at just half the rate. So the bias is 1/2. Or we can be more aggressive with a sample bias of 2 that really front loads the samples at the initial part of the FID. And I'd of course be remiss not to note that this process goes back over 30 years to this first example by Barna and Lowy. And so the question to ask is, does uniform sampling actually increase sensitivity? Now, that's not the only question you might want to ask about non-uniform sampling, especially for sparse sampling, but let's start here. And what we're going to do is look at this again with Monte Carlo simulations. We're going to generate the time domain FID using exponentially biased sampling schedules with varying bias in Tmax. We'll add Gaussian white noise, and then we'll fit the time domain data to a decaying exponential. Then we're going to repeat that 10,000 times, histogram up the results, and display the knowledge relative to uniform sampling with a Tmax of 3T2. And perhaps the most interesting parameter is going to be the signal amplitude. And that corresponds to the peak area and the frequency domain. And it answers the important question of whether a peak is present. So what I've plotted here is the knowledge of the amplitude for various biases and various Tmax. And they're all plotted, or their values are all listed relative to uniform sampling out to 3T2. So that has a knowledge of 1. And all the points correspond to equivalent 2D acquisition times, and the higher points correspond to better knowledge. And so from this, you can see that NUS is definitely better than uniform sampling at defining the knowledge of the peak amplitude. And in fact, for all the sampling schemes and biases, the best way to go for a finite number of samples is to put them earlier in the free induction decay. Now, of course, the other peak parameters of line width and frequency require the signal at other times. And so what I show here is the relative knowledge for those parameters as well. And indeed, more aggressive biases that work well for amplitude tend to show decreased knowledge for the line position and the line width. But if you want equivalent knowledge in position and width to say the uniform sampling, then perhaps using matched NUS, so a bias of 1 and a Tmax somewhere between 1.5 and 3 is a good way to go. Okay. When you have 2 peaks present, the choice of bias depends on the required resolution. So if the peaks are well separated, here separated by 5 line widths, then the knowledge improvement mirrors what we saw for a single peak. And you can decide how much information are you willing to give up in position and line width to get better knowledge of whether the peak is there or not. But as the peaks get closer together, much of that advantage of non-uniform sampling is lost. But importantly, matched NUS never falls below uniform sampling. And I should point out, the knowledge loss here isn't inherent to NUS. It actually has to do with the loss of resolution. And what I'm trying to do is separate the amplitude and positions into the individual peaks. If I just forget about that separation and look at the sum amplitude, well, the sum amplitude looks almost identical to what we're seeing for the well-spaced peaks. So this is a question of the assignment to the individual peaks due to the loss of resolution. Okay, so let me make a midpoint conclusion here. That there's absolutely no disadvantage to using NUS, and in most cases, it's considerably better than uniform sampling. So we definitely all should be doing NUS, provided that we have a way of constructing a frequency domain spectrum that preserves all of the knowledge gains. And so a number of techniques have been put forward to transform NUS data into the frequency domain. And they tend to be fairly robust, but they're often, and not always, nonlinear. And because of that, they lack the fundamental correspondence between the 2 domains that's conferred by the Fourier transform. And we saw earlier that this guaranteed that the fits in the time and the frequency domains preserve spectral knowledge. Brief shout out, however, some of these techniques have tried to address this. For example, MINT is operating in a linear regime and does, for uniformly sampled data, reproduce the Fourier transform. Today, I want to focus on these bottom 2 techniques that I'll show you guarantee the preservation of spectral knowledge, at least when they're suitably modified. And the first is an older technique, non-uniform weighted sampling. And the second is a new technique that we've recently introduced, called the block Fourier transform. Now, both of these are applicable only to relatively dense data. So it's not a full solution to the problem, but will be of use when you have sensitivity limited experiments. Okay, under non-uniform weighted sampling, we take multiple samples at each time point on the Nyquist grid. And we do that with some bias, typically exponential. And then these replicate time points are just summed to give the NUWS time domain signal. And because this is on the Nyquist grid, you can just Fourier transform this into the frequency domain. And so this seems like a really simple way to do NUS. So what does it do to spectral knowledge? So again, we can run Monte Carlo simulations, comparing the NUS in the time domain to the NUWS procedure. So in the time domain, we just measure multiple points at each time point, and then we just fit this in the time domain. And in NUWS, we sum those, and we can fit that in the time or the frequency domain. It doesn't matter. They give identical results, as I showed earlier, because they're related by a fast Fourier transform. And then I just repeat this process 10,000 times for each, keeping track of the various parameters as I go through the Monte Carlo cycle. And when we do that, what we find is that NUWS, shown here, maintains about 80% of the potential increase in knowledge from NUS in the time domain. And that's not bad, but can we recover the full knowledge? And the reason why the knowledge is lost can actually be traced back to what happens to the noise when we make the NUWS signal. So here, each of these individual time points have identical noise associated with them. But over here, each of these time points no longer has the same amount of noise. This point here was constructed by the sum of 10 time domain measurements. And so it has root 10 more noise than, say, this point out here, which is just a single point. And so we can account for that if we're fitting in the time domain in terms of the chi-squared function that we minimize to fit this data. But if you're going to Fourier transform the data, you're going to lose the ability to make this simple assignment of the noise at the individual points. And that fundamentally is why when we Fourier transform the data or ignore the fact that these points have different noise, that we lose some of that knowledge enhancement. So the solution to this is actually straightforward. We just need to construct a signal in which the points all have the same root mean square noise. And this we call using consistent RMS noise, or UCR. And it's equivalent to dividing each of those sum time points just by the square root of the number of points that was used to form that sum. And so we call this NUWS UCR. And if we run the Monte Carlo simulations again of the frequency domain spectrum generated with NUWS UCR, we find that we can preserve the full NUS spectral knowledge gains. And that's true whether we fit it now in the time or if we fit it in the frequency domains. Okay, so NUWS UCR is a robust technique, but it still cleaves to the Nyquist grid. And so I want to introduce a second approach that also maintains the full NUS advantage, but allows you to go off the Nyquist grid and may allow us to go to slightly lower sampling densities. And this approach is called the block Fourier transform. And it's a fundamentally different way of viewing the FID, right? The discrete Fourier transform treats this signal as a set of impulse functions. And if you leave one of these out, or if you change the spacing, this gives you problems in the frequency domain reconstruction. So instead, I want to look at the FID in a different way. And what I want you to see is not the points in the free induction decay, but what I want you to see is the connection between the points. That is, I want to introduce the idea of the free induction decay as a series of line segments. And the reason I want to do that is because each of these line segments has an analytic Fourier transform. And so now in this model of the free induction decay, it's just a series of piecewise continuous line segments, and the Fourier transform can be written down, simply as the sum of those the Fourier transform of those individual lines. And in the limit of uniform sampling, this can reproduce the discrete Fourier transform, but it now allows us to go to general off-grid non-uniform sampling as well. Okay, so how well does it work? So again, we can do Monte Carlo. We start here with off-grid time domain NUS with a exponentially biased sampling density, and we can just fit that in the time domain, or we can use the block Fourier transform to generate the frequency domain and fit it there. And we can run these Monte Carlo cycles, again, 10,000 times, and compare the knowledge in the extracted spectral parameters. And again, it's a disappointment. In this case, an even bigger disappointment. The block Fourier transform only maintains about 40% of the potential increase in knowledge from NUS in the time domain. So yeah, it's just doing bad. And again, the problem is the weighting of the noise in the time domain signal before the transformation. Luckily, there's, again, a simple solution. What we need to do is weight the Fourier transform of each of those line segments by the square root of its length so that it contributes equivalent noise in the frequency domain. And once again, we call this using consistent RMS noise, or the BFT UCR. And when you run this through Monte Carlo, what you find is that the BFT UCR fully preserves knowledge in the frequency domain compared to NUS in the time domain. So now we have two methods that fundamentally preserve knowledge when going from time to frequency domains. In other words, you cannot do better than these two techniques. And we say that they provide or produce knowledge equivalent spectra to the time domain data. So I've been talking mostly about knowledge of spectral parameters, and I know that I can't escape questions regarding signal to noise ratio. Remember that these are actually tied. The knowledge and increased signal to noise ratio go hand in hand. Again, you have to be very careful to distinguish between the intrinsic or fundamental signal to noise and the apparent signal to noise. The apparent signal to noise is cosmetic. It doesn't necessarily correspond to increased knowledge, whereas the fundamental or intrinsic signal to noise mirrors the increased knowledge. Both the Fourier, the BFT UCR and the NUWS UCR show increased signal to noise ratios compared to uniform sampling of about 30 to 50%, just mirroring the gains on these plots. And this increase in signal to noise is intrinsic. It's not just apparent. Okay, so in the last few minutes, let me show you a few experimental examples. The first is an HSQC spectrum of tryptophan synthase at 600 megahertz. I apologize, Bob, I'm showing a solution state spectrum at your solid state Zoominar. And in this example, we're using matched NUWS on grid and then NUWS UCR processing using a Brooker Python script that Ivan Sergeyev wrote for us. And we see the expected signal to noise increase of about 25% in this case. It's not as large as the ones I was showing you earlier, because in this example, we're only going out to about 1.5 T2 in the indirect dimension. And if you check, okay, yeah, that's about the increase that we'd expect to see in terms of the signal to noise. Here's a second example from solid state NMR. One of the challenges in the reconstruction of NUS spectra is the large dynamic range and the presence of non-Lorentzian line shapes. And that's relevant here where we're looking at CSA correlation in this carboxylate labeled glycine sample. There's two magnetically inequivalent positions in the unit cell, and this 2D powder pattern just maps out the relative orientations of these two CSA tensors. And here we're using off-grid NUS and BFT UCR processing in NMR pipe. And I want to thank Frank Delagio for helping us put that together. In this case, the improvement's more substantial, about a factor of 1.7. And that's because we're actually going out significantly longer in the indirect dimension, based in part on just a really crude guess of what our T2 values were. Okay, so let me conclude by saying that Monte Carlo simulations are a robust method for assessing the efficacy of data acquisition and processing protocols. It turned me from a quiet skeptic of NUS to a true believer. And if one of your friends calls you up and says they have this great new acquisition or processing scheme that increases sensitivity, ask them to show you the Monte Carlo, because it doesn't lie. And so, also a really big conclusion is that we should all be using NUS. There's no disadvantage in terms of performance. And if you choose to use NUS, I hope you'll find the NUWS UCR and the BFT UCR might be helpful for you. They show both increased spectral knowledge as well as increased signal to noise ratios. And with that, I should acknowledge my collaborators on this, my graduate students, Manpreet Kaur, Callie Lewis, Dave Amarasinghe, Kevin Chilick, Jacob Holmes, and Chris Williams, my undergraduates, Aaron Chronister and Gabe Pham, and a special thanks to Frank Delagio, Ivan Sergeyev, Joachim Strupa, Dave Rovniak, and Jeff Hoke for very interesting discussions on non-uniform sampling. So finally, let me thank Bob and Kong again for putting together this spectacular series over the last year, and I'd be happy to answer any questions.
Okay, Lynn, really nice talk. I guess there are lots of skeptics of NUS, and still, so I have various friends who record protein spectra, and they always relax back to the uniform sampled things, because they tend to think that well, I don't know what their evidence is, but they tell me that the NUS schemes that they have available to them, I guess on their Bruker spectrometers, seem to generate some extra signal noise, extra noise, and lower the signal noise. But anyway, it would be good to try your schemes. Are they readily available on commercial instruments or even homemade instruments?
Yeah, so they are becoming available. So working with Ivan, we now have a really user-friendly NUWS processing script for the Bruker. It works in Topspin 4 right now, just needs minor modifications to work on Topspin 3.6. Those are critical because you're sitting at the spectrometer, you want to process it while you're there. That may not be the final way you process it, right? But you need to know that these things are working as you're acquiring them. And so that is quite helpful in that respect. Now, many of the people who are running protein samples are using sparse sampling. So they're going below 100% sampling, right? Even down to 10 to 20% sampling. These techniques that I showed you today will not address that. You can use Monte Carlo to assess how well those other techniques are working. And I would urge you to do that. But the techniques that I was talking about today are all for relatively dense or sensitivity limited samples. And there you have to take a little bit of a leap of faith. I mean, we do that in my own group. We're like, okay, is this the time where we just go NUWS and 4 days later hope everything is working perfect, when we can't see it, because it's sensitivity limited, we're not going to see it to the end. So you really have to put your trust into it. And I would say, yeah, it's time to do that. These work well. So grab your favorite sampling bias that will improve sensitivity, and you won't be disappointed.
All right, so we have some questions piling up here. So David Bryce asked, he said, you showed that acquiring extra points at the end of the FID doesn't hurt the signal. but then your graphs show decreasing knowledge as T over T, big T.
Yep.
Increases. I must have misunderstood something here, you said?
Yeah, thanks, Dave, for pointing that out, because that's a really important distinction. The first graphs were relevant for direct acquisition. Okay, so that is the case where taking more time points does not affect the overall experimental time. But of course, NUS in the direct dimension doesn't make sense. The points where the graphs I showed later was for NUS in the indirect dimension, where you have a finite number of samples and you're going to distribute them across your acquisition time. And so there, if you have 64 data points and you're pushing some of them really far out, well, then you're going to start to lose knowledge as you put the samples farther out. Now there's a sweet spot, if you'll notice. Let's see if I can get back to that. Right, there's a sweet spot for position and width where you've evolved far enough out that you can see the frequency evolve and see it decay. But then if you start putting your samples even farther out, you'll start to lose knowledge again. So it's the distinction between direct and indirect acquisition.
Okay. Yeah. All right, Lyndon Imsley says, nice talk, Lynn. You kept saying there were no disadvantages, but you did say there is a loss of knowledge on position when you gain knowledge on amplitude, didn't you? Question mark.
Yep. Okay. So you can so I guess this is a good curve to have up for that as well. Yeah, you're absolutely right. I keep saying there's no disadvantage, in part because so I misspoke. There is, but it's trivial, I think, in terms of most experimentalist considerations, to lose 10% knowledge in a central frequency that we already know to several hundreds of hertz is probably not the largest loss compared to increasing your signal to noise by 60 to 70%. So you're right, you do give something up, but it's not a lot. So thanks.
And so Walt wants to know, is there a minimum absolute signal to noise ratio that is necessary for the knowledge advantages of NUS to persist?
Well, that would depend on the processing scheme, and the schemes that I showed today, no, because of the use of, because both of the techniques I showed today get us back to a position where we can do a Fourier transform. Okay, so that correspondence between time and frequency domain and the linearity of the Fourier transform make these really robust techniques. So there's no minimum requirement for signal to noise, and you should benefit in all cases. Now, other processing schemes may have challenges, and so I can't answer that question for other processing schemes. And the processing for sparse sampling is extremely good. We've taken a look at some of them, and they do a pretty good job of retaining knowledge. They're not perfect from what we've seen, but we haven't tested them fully, I would say. And how they would respond to low signal to noise, I'm uncertain.
All right, and Brian Michael, as one of the concerns we have had using NUS is the presence of artifacts and distinguishing them from real signals. This is especially concerning when you have a strong diagonal and weak cross peaks. How well do you preserve knowledge when you have such big differences in peak intensity and the potential of artifacts on the scale of some of the real peaks?
Yeah, so if you're in a sensitivity limited regime, then I think you should try the NUWS UCR or BFT UCR, because they can handle large differences, just like the Fourier transform in intensities. That's a challenge for reconstruction in general. and certainly one that all the developers of the reconstruction are aware of and try to make their techniques robust to. One of the advantages in the BFT UCR and the NUWS UCR is that what you see for signal to noise is real. So it's not signal to artifact like you would see in many reconstruction techniques. What you see is the fundamental noise and the intrinsic signal to noise. And so you can sort of judge by eye and experience, what's peak, what's not. Whereas in the other reconstruction techniques, you're right, you lose that noise floor, and sometimes there might be small processing artifacts and you have a hard time distinguishing those. What I will say is if you do Monte Carlo on those processing techniques, you'll be able to characterize them, but that's probably not what you're looking to do when you have an actual protein spectrum in front of you. So if your sensitivity limited, try this approach and you won't have artifacts.
All right.
Well,
That's the bottom line. Okay.
Yeah.
Ben Wiley wants to know, is BFT UCR available in the current pipe release?
Yeah, so that's pretty much hot off the presses from the last couple weeks. So we're working with Frank and hopefully it will be available in an upcoming release. But right now, what is available, it would be the NUWS processing on Brooker. So that's a Python script we can send out now. It's still, it's not, the processing is user friendly, but you have to set up the NUWS. And so there's a few tricks to that we're documenting in a paper that we're writing right now, but I'm certainly happy to make you aware of to help get those set up easier.
So Thomas Schmidt says, I'm coming from the EPR Deerfield, and a large portion of our signal arises from an exponential background function, which is US, and which is corrected by subtracting or dividing the signal, which causes trouble with the NUS. If I understand correctly, your BFT can directly take care of underlying artifact as it only concerns corrected distance in the lines and the sample points in the time domain. And he wants to know if this is correct. That's a rather long question. Can you maybe?
Yeah, I think I'd probably have to discuss that in more detail with him. So I'd be happy to converse on that, but I'm not sure I caught all the subtleties there. And one of the take-home messages, hopefully from today's talk, is there's a lot of subtleties and they matter. So I don't want to misspeak on that, but potentially it could be applicable there. Now, realize that, in all of, when I kept saying, relatively dense data, what I'm really saying is that the data needs to be bandwidth limited. So if your frequencies are going faster than the spacing between the points, obviously these techniques, just like the fast Fourier transform, wouldn't be able to capture that. So as long as you're staying within that limit, there's the chance that the BFT should work for you.
All right, and an anonymous attendee has the question, can I say zero filling doesn't increase absolute signal to noise ratio?
Can you say what?
Can I say that zero filling does not increase absolute signal to noise ratio?
Oh, that's interesting. I mean, that could be a talk on its own. And if you'd given me an hour, I would have addressed that, Bob. So interesting thing about, so go do the Monte Carlo. It's really fun to see. If you zero fill, you don't actually change the signal to noise. You just double the points in the line. And so you get √2 better signal or better parameter estimation by doing that because you've doubled the points. What's critical though is when that first zero filling actually increases the number of independent points. If you zero fill any more than that, you're just extrapolating points. It doesn't help you. But the first zero fill, because the real and the imaginary components are Kramers-Kronig related, that first zero filling actually takes information that was in the imaginary domain and moves it into the real domain. Said another way, if you're to fit your spectrum and just fit the real part, not the real and the imaginary part, and then do zero filling and just fit the real again, you would see root 2 better fit parameters. If you're fitting both the real and the imaginary simultaneously in all cases, you wouldn't see any improvement from zero filling. So it doesn't actually change the signal to noise, but it does increase the density of independent points in the real dimension. That's a fun talk on its own though. So that could.
Yeah. Okay, so I just received an email from David Rovniak, one of your collaborators. And he says, a quick follow-up. Yes, Bruker-generated schedules should never be used and have caused a lot of bad impressions. Poisson gap is improved over the poor Bruker schedules, but has several weaknesses. He says, I will help generate robust schedules for anyone interested. If you have someone in your group who wants to generate good NUS schedules, I can train them to make good schedules. This worked recently in our collaboration with Gary Martin, where once they started using good schedules, they were impressed with NUS. Also, I know how Lynn generated his schedules, and he did a really great job, of course, so either of us could help generate good schedules. Anyway, that was David's comment.
Yeah, I fully endorse Dave's schedulers. They're great, they're robust. We do all of ours, we just do ours in Mathematica and put them out as text files that you can read into the Bruker.
Yeah. Okay.
I think Chen has a question. Chen.
Oh yeah. Yeah, nice talk, Len. So a quick question, you may have mentioned, but so for the Monte Carlo simulation to evaluate the efficiency of this method, so or effectiveness, so what kind of distribution you were drawing sample from? Was that a normal distribution?
Yeah, it was just Gaussian white noise. So it was a normal distribution. I didn't show the plots, but we did double check that so we were doing all this in Mathematica, and we did double check the distributions were in fact Gaussian. We did check the convergence of the Monte Carlo, and by 10,000 cycles, we're extremely well converged.
Nice. Yeah, thank you.
Yep.
Okay. Well, there are no more questions, and it's now 12:30. So I want to thank again Andy and Lynn for really 2 very interesting, inspiring talks today. Lots of new information, new approaches to dynamics, as well as to non-uniform sampling. So those are 2 really important topics for the world of magnetic resonance, both EPR as well as NMR as well as EPR. All right. And so, Kong, do you have an announcement or schedule for
Sure.
2 weeks from today?
Yeah, 2 weeks from today, like there will be 20th of April, we will be hosting Harmo Oskina from Berlin and Vlad Ladistansky. I don't know how to say his name, from University of Guelph.
Okay.
Yeah.
So we will see everyone in 2 weeks. And again, a big thanks to Andy and Lynn for 2 really great talks. Okay. And see you again in 2 weeks, we hope. Bye.
Bob, shall I see you again at 2:30 later?
Good. Okay. All right.
2:30, right? Okay.
Okay.
All right. Bye. Thanks, Andy. Bye, Len.
Thanks.
Thanks.
Bye.
Thank you.